Hi, everyone. I'm a fourth year biology major. After I graduate from my undergrad, I'm going to go on and pursue my PhD in plant bio here at UCR. And currently, I'm under the Rasmussen lab. And today, I'll be presenting on the characterization of Tangled 62. Okay, so to start off, our lab studies division plane orientation. And division plane orientation can be described as the final position in which the cell places its cell wall. And so in a quote unquote correct division, you have the cell establish a predicted division site early in mitosis. And at the end of mitosis, you have the cell place its cell wall on the predicted division site. However, there are some instances in which cells undergo a misoriented division, and a misoriented division can be described as the cell wall being placed away from this predicted division site at the end of mitosis. Okay, and so the reason that we study division plane orientation is because it's important for proper growth and development across many different organisms. And so here you have an image of maize, and this plant right here shows a plant with no division plane orientation defects. And as you can see here, it grows to its full potential. However, in this plant right here, you have the division plane orientation defects, and the plant is significantly stunted in growth. And at the cellular level, division plane orientation defects causes misoriented divisions, which you can see here that the cells lose the brick-like structured patterns that are seen in the wild type plants. And so this is also important to animals because division plane orientation defects can cause a cell division that leads to the daughter cell breaking off from the substrate. Then the cell goes into proliferation and turns into a tumor. And this can also be seen in cancer metastasis. And so overall, studying division plane orientation can further our understanding about proper growth and development across many different organisms. Okay, so with the intention of our lab being interested in division plane orientation defects, our lab did an Ems Mutagenesis experiment, and through forward genetics, we were able to identify a new mutant called Tangled 62. And Tangled 62 was identified through glue impressions, which is using glue to take or to be able to visualize the cells in the epidermal tissue. And so as you can see here, Tangled 62 shows a lot of the misoriented divisions compared to a leaf that of a wild type plant. And so this mutant was known to have is known to have an asymmetric cell division defect. And so in asymmetrically dividing cells, you have a subsidiary mother cell divide to form the subsidiary cell and the pavement cell. And again, in these cell divisions, you again see the predicted division site, which is the location in which the cell wall will be placed. And this structure is a microtubule structure, which helps lay down the cell wall. And this is a movie taken by Lian Hong of a wild type cell undergoing mitosis. And as you can see here, the cell wall is placed at the correct division site. However, in cell division defects, you have the predicted division site. However, the microtubule structure is misguided to a different location, and this causes the cell wall to be placed away from the predicted division site. And this can be seen in a mutant called opaque 1. As you can see here, the cell wall is placed away from the division site. And so Using a microtubule marker line, we were able to image TAN62 using confocal microscopy. And this image just shows the microtubule structure being misguided to a different location than the predicted division site. And this shows a wild type cell that shows where the cell wall should be placed. And so when we were looking at the characteristics of Tangled 62, we noticed that the phenotypes are very similar to an already identified mutant called opaque 1. And so we went ahead and did a complementation test in which we crossed the Tangled 62 mutant with an opaque mutant. And we were expecting the different mutants to complement one another. And so we were expecting to observe the wild type phenotype, which is shown here. However, when we crossed these two, we observed the mutant phenotype, which is the opaque kernels shown here, the stunted growth, and the asymmetric cell division defect, which can be seen through glue impressions. And so learning more about opaque 1, through known literature, we were able to find that it encodes a myosin 11 protein. And this is a representation of how myosin 11 walks along the actin filament using ATP hydrolysis. And so it is known that opaque 1 has, again, the opaque kernel phenotype and the phragmoplast guidance defect. And it's a loss of function mutation. And this mutation is an adenine guanine deletion in exon 14, and it causes a frame shift and a premature stop codon. And so since Tangled 62 and opaque are so similar in phenotypes, the reason that I'm interested in learning more about the Tangled 62 mutation is because it has a more severe phenotype than the opaque 1 plants. And so this schematic is shows the opaque 1 gene, and the arrow points to the site of the location, which results in the opaque 1 phenotype. And this is a figure representing the myosin 11 protein with the different domains that serve different functions. And so one possibility that we've come up with is for why the Tangled 62 mutants have the more severe phenotype is that there's a single nucleotide polymorphism, or a SNP, that's leading to missense mutation in the head domain of the protein. The head domain of the protein is a site where binding to actin occurs, and this more severe phenotype may be causing the myosin 11 protein to capture an interactor and not deliver it to the correct site. And so in order for me to find exactly what is causing the TAN62 mutation or the phenotype, I am trying to amplify the opaque 1 locus in Tangled 62 mutants to be able to identify exactly what the mutation is. And so far, I've been able to design multiple sets of primers that span the entire opaque 1 gene. And then I went ahead and extracted RNA from Tangled 62 mutants and tested them for their integrity. Then using the best samples, I went ahead and did cDNA synthesis. And right now I'm at the part where I'm trying to amplify the gene. And so I'm working on troubleshooting to find the best ideal conditions where I can get rid of nonspecific binding. And once I get the expected band size with no nonspecific binding, I can send out the samples for sequencing so I can find exactly the location and what mutation is causing the TAN62 phenotype. Okay, I would like to thank the Rasmussen Lab and Carolyn, Lindy, and Stephanie for guiding me through this project. I appreciate you guys' help, and I would also I'd like to thank USpark for funding my research.
All right, excellent work. We got all of our round of emoji claps. Great job. Any follow-up questions? Yes, it's Dr. Brailsford. Go ahead and unmute. Oh,
Sorry, I was muted.
there you go.
What methods are you planning to use to solve the problem of non-specific binding to amplify the gene?
So I'm planning to, well, I've done a PCR gradient where I'm testing different annealing temperatures to see what the best annealing temperature is to get rid of the non-specific binding. But from there, I'll probably maybe change the actual master mix. Yeah.
Thanks.
Any additional questions? All right. Well, seeing no additional questions, Gabriela, you can go ahead and hit stop.